Welcome to Kai's Visual Biology. Today, let's talk about DNA replication. The first step is the unwinding of DNA's double helix structure, which requires a special enzyme called helicase. If you imagine yourself as a helicase, here's what happens. On the right is the front view. You go along the double helix structure. On the left is the rear view. You can see previously attached complementary nucleotides separate and form two single strands. The upper yellow strand, which goes from 3' to 5', is called the leading strand, and the lower orange one is called the lagging strand. Let's go with the leading strand first. In this process, RNA primer, the pink thing, is used as the starting point. As another enzyme polymerase goes along the leading strand, free nucleotides come to form another strand. Finally, polymerase reaches the end. Nucleotides replace the RNA primer and completes the replication of leading strand. Things get complicated with lagging strand because nucleotides can only form a strand from 3' to 5' sequentially. The replication goes backwards on lagging strand. Multiple RNA primers, starting points, start the replication of Okazaki fragments backwards. Again, nucleotides replace RNA primers, and another important enzyme, ligase, comes to bind fragments together to form a new complete strand with the template of lagging strand. To conclude, DNA replicates itself through semi-conservative replication and produces two identical copies each time. The study of DNA, or genetics, dates back to 1857, when Gregor Mendel performed his famous pea experiment. Later, Rosalind Franklin used X-ray diffraction to determine the three-dimensional structure of DNA. Then, James Watson and Francis Crick finally came up with the double helix model of DNA molecule. Currently, scientists are focusing on analyzing the human genome so that they can help people avoid some deadly diseases. The study of DNA answers the philosophical question, who am I and where do I come from, in a scientific way.